Hey, hi everyone. It's really nice to be here. Really happy to have the opportunity to tell you a little bit about what we've been working on at Sania, and in particular our lead therapeutic program, which is a targeted and controllable gene therapy for spasticity. Normally when I introduce Sania and what we do, I talk about how we're interested in nervous system disorders, and I have to immediately stop people from thinking about things like Parkinson's or Alzheimer's or classic neurological conditions. So I was really happy to see in the previous session being pointed out that the nervous system actually controls a whole range of different processes. So everything from blood pressure, circulation to hormone levels, and as well as motor output, are affected by really discrete and known populations of neurons within the central and peripheral nervous system. Ultimately, that's one of the things that is obviously disrupted in spinal cord injury and why there's such a range of different symptoms for patients. So what we're trying to develop at Sania are gene therapies that can take advantage of the nervous system and these neural circuits that control these different body processes. We're trying to target these very precisely with viruses at very low doses that have very clear efficacy for therapeutic effect. Now, also normally when I explain what Sania does, I have to spend some time talking about what spasticity is, but I think for this audience it can be a little bit more brief. But I'm always surprised by the relative lack of knowledge of spasticity, given that it's a really highly prevalent movement disorder. So I'm sure you all know there's around about 12 million people in the world who suffer from spasticity, not just from spinal cord injury, but also from things like stroke, cerebral palsy, etc. All cause these really painful, uncontrollable muscle contractions that characterize spasticity. Of those 12 million, in the UK, US, and EU, around about 2.5 million suffer from really severe forms of the disease, which are not controlled well with current therapies. Now, when you talk about gene therapies, people immediately start to think of very expensive, $2 or $3 million treatments for things like hemophilia. That is ultimately not what we're trying to build at the moment. What we want to do is build a gene therapy that's accessible to as many different patients as possible. So on the right of the slide here, it's a snapshot from a market analysis we had done, which sets the price point of a gene therapy at something that is relatively affordable given the current standard of care in spasticity, and just shows that if you have these large patient populations, such as in spasticity and other disorders, you can make gene therapies affordable for large patient groups, and that's really what we're trying to do. Now, spasticity itself, just to talk about the actual causes of these severe muscle contractions. Now, even although things like spinal cord injury, stroke, cerebral palsy, etc., all have different upstream causes and cause different things to the nervous system, ultimately what causes the spasticity symptoms is disruption in one very particular neural circuit in the spinal cord. And I'm sure you're all familiar with spinal motor neurons, but just in case, these are the motor neurons that sit with their cell body in the spinal cord, send their axon, their projection, out to the periphery. They contact muscles. When that motor neuron fires, that's how you contract your muscle, and that underlies all the different movements that we make. In spasticity, really specifically, those motor neurons become hyperexcitable. So they're too electrically active. They respond too much to other neuronal inputs, to other sensory inputs, to other descending inputs from the brain, and that's ultimately why they cause these uncontrollable muscle contractions. So what we're trying to build at Sania, what we are building at Sania, is a two-part gene therapy that will control these spasticity symptoms. The first part is an engineered adeno-associated virus vector. So this is a safe viral vector that's used currently in gene therapies. And this is injected directly into the spastic muscle. That is not to target the muscle itself, but it's designed in order to target the terminals of the motor neurons that are causing the muscle contraction. So it binds to the terminals of the motor neurons, is transported into the central nervous system, into the spinal cord, and then expresses a particular ion channel within that motor neuron. On its own, this ion channel is relatively inert. It doesn't change the physiology of the motor neuron, but it does make those motor neurons selectively susceptible to a particular small molecule that the patient takes orally. When that small molecule is taken, it binds to the motor neuron and dampens down that hyperexcitability. And that, we think, gives us the advantage that you can titrate the level of that small molecule in order to control spasticity, which, as we heard earlier, is very variable according to many different factors. So to get this to work, we need to do two things, basically. We need to be able to precisely deliver that ion channel just to the motor neurons with as few off-target effects as possible. We do that by engineering these adeno-associated virus vectors. And then we need a neuromodulation system that ultimately controls that hyperexcitability. So I'm going to talk you through the two halves of this therapy. So first of all, for the precision delivery aspect. Now, in gene therapies, these viruses, these AAVs, have been used quite widely for a variety of different conditions. And now what people are starting to do, and have been doing for the last few years, is engineer novel viruses that bind to particular cell types, so have a cell type specificity. One of the real problems that has occurred in the field is you can evolve these new types of vectors in animal models, so mouse or non-human primate, but it's very difficult to predict the translation of those vectors into humans because they have very cell type specific properties. So what we do at Sania is we take a slightly different approach where what we do is a human first approach is what we call it. So basically our first step of our R&D platform is we try and rebuild some of these neural circuits that are disrupted in things like spasticity, so these motor neuron to muscle circuits, say. Build those using iPSCs, using stem cells, in a proprietary microfluidic system. We then use those to evolve new AAV vectors that can target, we think, motor neurons. We then test and validate those vectors in animal models, so mouse and non-human primate. And what we're really looking for there is consistent properties, consistent tropism of those vectors across both human in vitro screens and mouse and non-human primates. So ultimately, they have predictable properties, hopefully, when they get to the clinic. I'm only going to show you, I think, one data slide or two data slides on this precision delivery system. So I think we're all familiar with the spinal cord. One of the advantages excuse me, of the spinal cord is the spinal motor neurons are well organized within the cord. Motor neurons which connect to one particular muscle are found in particular clusters called motor pools. So these are experiments where animals, mice, have received a single intramuscular injection in their calf muscle of either a wild-type vector, so the kind of current standard gene therapy vector, AAV2, or a couple of R vectors that we've engineered in the lab. So looking at the red images in the middle, the first column is AAV2. So after you inject into this particular muscle, you don't see any motor neurons labeled. So it's very difficult to target motor neurons via an intramuscular injection. With R, and that's kind of quantified just on the right with the green bars that you can barely see, because it's really just 1 or 2 cells per animal that you label. With R vectors, what you do get, so numbers 12 and 43, shown in the middle and on the right, you get almost all of the motor pool that's innervating that muscle. So around about 80% of the motor neurons that innervate that muscle are transduced with R vectors. And we think that's much more what you're going to need for therapeutic efficacy. And that's really important because that allows this minimally invasive intramuscular injection to prime the motor neurons for the small molecule. Obviously, we're very interested in the safety of these vectors. And we want to know, if we're going to use these vectors, where else in the body do they end up after this intramuscular injection? So these are some biodistribution studies, again, injecting groups of these vectors mixed together into the same calf muscle of a mouse. And then we're just using qPCR to find out where the vector has ended up in off-target in other tissues. And the reason for that is one of the drawbacks of gene therapies has that you see many of these vectors taken up in things like the liver, heart, etc. And that can cause all sorts of toxicity. When you do an intramuscular injection of these vectors, in our hands, you don't get any expression in off-target tissue. So really everything stays within the muscle, within the motor neuron, but nothing ends up in the liver, heart, or other tissues that you would worry about. So I'll talk about the second half of the therapy, how do we control the excitability of these motor neurons. So we can deliver the gene using these AAV vectors that we've engineered. And what's important to note is that this is a one-time injection. So that gene should stay on, ideally for the lifetime of the patient. So it's just a one-time intramuscular injection. What the patient would then take is this oral small molecule once or a couple of times a day to dampen down excitability. So the AAV drives expression of a human ion channel and basically overexpresses that ion channel that is found in motor neurons. What that means is that when you take a very low dose of this small molecule, it selectively activates these motor neurons and dampens down their excitability. And I'll show you just a couple of data slides demonstrating that. So first of all, these are electrophysiology traces of human motor neurons in culture. So this is recordings using something called a multi-electrode array. It's not particularly important, but these are networks of human motor neurons found that we generate in a dish. They're all active, electrically active, talking to each other, and you can record that spiking activity of those motor neurons. We measure many, many different properties of them and have done many different experiments, but just showing you a couple here. So the center graph in pink, which you can barely see in the center, and gray. Pink is neurons which are overexpressing this particular ion channel, and gray is wild-type neurons. And what we're measuring here is the interaction between overexpressing that ion channel and applying our small molecule drug. And what you can see, I hope from the graph, is that there's no change in viability. So that means the cells stay very healthy as you apply this combination. On the right, though, what you can see is how the number of action potentials that these neurons generate change when you apply the small molecule. So again, pink is overexpression of our channel, which we call C490, compared to wild type. And you can see that very, very low doses of this drug, and this is a clinically approved, widely available drug, that low doses of these of the small molecule drives down the excitability of the neurons really potently compared to control. So we think this is a clear way, a clear efficacious way of driving down motor neuron excitability. Finally, we've validated this in an animal model of spasticity, in this case in a spinal cord injury model. So this is a neonatal mouse spinal cord injury model. Animals receive spinal cord injury at neonatal ages. They're allowed to grow up, and they develop spasticity at a few weeks after birth. So here what we're doing is we're injecting either a control vector expressing an inert protein, GFP, which is just a fluorescent protein, or our particular channel, the C490. So you can see, I think, from the graph on the left, when you have just this inert protein, you apply this small molecule activator, you don't get any change in this particular measure of spasticity, which is the duration of muscle spasms, measured by EMG, which is one of the same ways you would do in human patients. When you use this combination of overexpressing our channel and adding this particular activator, a small molecule activator, you get a clear reduction in spasticity. So we think this is quite an effective way of reducing spasticity symptoms, and that's kind of shown with some example EMG traces on the right there. So very briefly, I'll just tell you kind of where we are and what we're thinking about in terms of our pipeline, just in the last 2 minutes. So spasticity, as I said, is our lead program. We are currently moving that vector validation into large animals, into non-human primates. And our plan is to do formal toxicology studies next year, aiming to be in the clinic in 2026. We have other SCI-relevant programs, particularly for things like neurogenic bladder, which again is underlined by hyper-excitable motor neurons. So we think the same kind of product, same lead candidate that we developed for limb spasticity can be used for things like neurogenic bladder. So finally, just highlight my team. I'm around for the next day or so, happy to answer a couple of questions now, I imagine, and any other questions that you might have over the networking sessions. But thank you for your attention.
Great, thank you so much for the presentation. This first question is a bit high level, but it seems that your intramuscular injection is very much localized to the point of injection. How do you, I guess, envision translating this to humans who maybe have larger muscles? And do you imagine multiple intramuscular injections, or is it a singular site that you're going to inject the therapy at?
It would be multiple injections into one muscle. It would be relatively similar to Botox injections that you might have for spasticity already. One of the advantages in humans, as opposed to mouse, is we know exactly where the synaptic terminals are in each muscle, so you can target basically the injections to the right place in order to get to the motor neuron. So you would have to have multiple injections, but it is just a one-time procedure.
Got it. And then the other question I wanted to ask was around the cost of the therapy. Those that follow the gene therapy space may be aware that these are, I guess, very expensive therapies. There was a drug that received an expanded approval for Duchenne muscular dystrophy last week that's going to cost $3.2 million per patient. How do you think about combating the high cost of these therapies? What are you doing on the manufacturing side to ensure that you're able to produce this in an efficient manner?
Yeah, it's a question we get a lot. I think we have two advantages. So firstly, because we're doing this localized injection, the dose that we need is at least 100, probably 1,000-fold less than you need for more traditional, say, Duchenne's or hemophilia. So the cost of goods for manufacturing the AAVs are, much, much, much lower. So that allows us immediately to drive down the price point. And I think also just having a really large patient population allows us to have a much more reasonable price point than 2 or 3 million. I don't think that's really not what we want to do. We want to get as many patients as we can, or help as many patients as we can.
And maybe one more quick one. It seems like you're working on novel vectors. What's your expectation as it relates to immunogenicity of those vectors in humans? That's been an issue as well, enrolling patients in these studies.
Yeah, it's a very good question. So I think you're alluding to the fact that if you have antibodies already to particular AAVs, you can't have the gene therapy. So our hypothesis, and I would say we've not tested it directly, but we will do in the next 6 months or so, is that if you're doing an intramuscular injection just to target the terminals, actually the immune response is going to be relatively both minor and non-consequential, because you're really injecting at the point where you're transducing the cells. So there's not going to be enough time for circulating antibodies, for example, to come and break down the virus. But we're testing that at the moment, but also because they're novel vectors, we're hoping that they won't have the same pre-existing immunity in the population as you get with other gene therapies.
Got it. Great. Thank you so much.
Thank you.